Hello everyone. Today we are going to discuss about the topic electron transport chain. What is electron transport chain? It is the transfer of electron from NADH or FADH to 2O2. The electron flow occurs through the successive dehydrogenase enzymes together known as electron transport chain ETC. Site of electron transport chain. Electron transport carrier protein complexes are located in the inner mitochondrial membrane. Inner mitochondrial membrane is folded into the cristae. Density of cristae in the mitochondrion is related to the energy requirement of the cell. Components in electron transport chain. There are 4 components in electron transport chain. They are electron carriers, mobile electron carriers, ATP synthase and oxygen atom. Electron carriers. Electron carriers of the electron transport chain are located in 4 complexes. Complex 1, Complex 2, Complex 3 and Complex 4. Complex 1. NADH dehydrogenase. It consists of 2 prosynthetic groups, flavin mononucleotide and iron sulfur center. Electrons from NADH are transferred to the complex 1 containing FMN and then to the iron sulfur centers. Then the electrons move to the mobile electron carrier called coenzyme Q. The complex 1 pumps 4 H plus ions to the intermembrane spaces of mitochondria. Complex 2. Succinate dehydrogenase. This complex includes the enzyme succinate dehydrogenase, the same enzyme that participates in the Krebs cycle. Prosynthetic groups present. Flavin adenine nucleotide FAD and FES centers and heme. Path of electron transfer. Electrons from succinate transfer to FAD, then to the FES center, then to the cytochrome B. Complex 2 does not pump H plus ions. Complex 3. Cytochrome B and cytochrome C1. This complex is named as cytochrome oxidoreductase. Heme is the prosthetic group of cytochrome B and cytochrome C1. It is also consists of FES centers. Path of electron transfer. Electrons from coenzyme Q are transferred to the cytochrome B, then to the cytochrome C1. It pumps 4 H plus ions to the intermembrane space. Complex 4. It is the last complex for the transfer of electron. Prosynthetic groups present. It consists of cytochrome A and A3. Moreover, it possesses copper A and copper B. It pumps 2 H plus ions to the intermembrane space. Next, mobile electron carriers in ETC. They are able to physically transfer electrons from one site to another site. There are 2 mobile electron carriers in ETC, coenzyme Q and cytochrome C. Coenzyme Q. It is also called ubiquinone. Function of coenzyme Q. It transfers electrons from complex 1 to complex 3, also transfer electrons from complex 2 to complex 3. Cytochrome C. It is a soluble protein which binds on the mitochondrial membrane to perform its electron transport function. It plays a role in programmed cell death. Function. It transfers electrons from complex 3 to complex 4. Functioning of ETC. In electron transport chain, the constituent molecules are arranged in the order of increasing redox potential, decreasing electron pressure, diminishing free energy. So, along with the chain, there would be step-by-step flow of electrons from most negative initial donor to the positive terminal acceptor. Electron entering ETC are energy rich. As they flow down, they lose free energy. Much of this energy gets conserved in ATP. In electron transport chain, enzyme bound H is used as fuel of energy generation. Available form: the dehydrogenation reaction of substrate oxidation. From H donating substrate, H atoms are collected by dehydrogenase and supplied to the ETC through NAD and FAD. They are serves as the acceptor, carrier and donor of H. The H atoms soon donate their electron to the electron transport chain. These H plus ions soon escape to the aqueous medium. NAD linked dehydrogenase remove two H atoms from the substrate. One transport to the NAD as hydride ion. One appears in the medium as H plus. FAD linked dehydrogenase remove two H atoms from the substrate. In most cases, both of them will be accepted by FAD and yielding FADH2. From the coenzyme channel, electrons are funneled to molecular oxygen through the channel of electron carrier. They include flavor proteins, iron sulfur centers, ubiquinone, coenzyme Q and cytochromes. During the flow of electrons, every members of electron transport chain get alternatively reduced and oxidized in cyclic manner. In this process, iron atoms oscillate between Fe2 plus and Fe3 plus states. At the initial end, the redox reaction chain is linked to the dehydrogenation reaction and at its terminal end to the molecular oxygen. At the same time, some of its intermediate reactions are coupled with the phosphorylations of ADP. The proton tend flow back to the mitochondrial matrix through ATP synthase enzyme complex. The release of energy that happens when protons flow down the concentration gradient to the matrix permits the ATP synthesis of enzyme complex to the phosphorylate ADP to ATP. This process is called oxidative phosphorylation. So, it is a functioning of electron transport chain. Thank you.